All right, here we are to talk about the rough and smooth ER and Golgi. So the ERs are just a bunch of membranes or sacs that are attached. So it is a network. So you can imagine it's like a bunch of hallways that you could walk through if you were small enough. And it's going to provide mechanical support. And what that means is it's going to provide support for the cell in the way that the cell works to allow it to withstand any harsh or extreme conditions. And so it's going to help the way the cell functions so that it can handle environmental conditions that change. It also plays a big role in intracellular transport. There are two types. There is the rough ER, and the rough ER is called that, as you can see, because it has little ribosomes all over it. And the smooth ER, which generally takes on a more tubular shape, more tube-looking, and it does not have any of the ribosomes. And this is an actual micrograph, so a picture from a microscope, probably taken with a transmission electron microscope. And you can see the rough ER has the ribosomes attached, and the smooth ER does not. All right, so both of them compartmentalize the cell. The function of the rough ER, main function is to make proteins. That's why it has ribosomes. And those proteins are going to be sent generally to the Golgi and then out through vesicles to the cell membrane for exocytosis. So generally making proteins that are leaving the cell. It can also send other things. So if it has something or if it receives something that needs to go either out of the cell or just somewhere else in the cell, it can package that up and send it to the Golgi. And it also is one of the organelles responsible for replacing membrane that's been damaged, whether that's membrane on another organelle or whether that is membrane for the actual cell, or if a part of the membrane is just getting old. So it's just like your skin. You're constantly replacing old and worn out skin cells, and your cells are doing the same thing with their membrane. The smooth ER, again, it's smooth because there's no ribosomes. It has a bunch of different functions. It makes lipids, so it's producing the phospholipids and all the other lipids that your cell needs. It's going to break down carbs, so it's metabolizing your carbohydrates along with your mitochondria. It's probably breaking them down, or it is breaking them down to make it easier for the mitochondria, because mitochondria really prefers carbs in their broken down form. So this is kind of like chewing them up further to get them ready for the mitochondria. It also detoxes drugs and poisons, and this is one reason why we see when you look at the liver cells of an alcoholic versus someone who's not alcoholic, it is why you see more smooth ER in their liver cells, because they are putting more poison into their body, and so their body is trying to speed up the process of breaking that poison down. The other thing the smooth ER does is it stores calcium. This is really important for a lot of things, but one main thing it's important for is the release of neurotransmitters in your neurons. And so the smooth ER, when a neurotransmitter gets an action potential and needs to release when a neuron gets an action potential and needs to release a neurotransmitter, it will, part of that action potential is telling the smooth ER to release calcium. That calcium will then cause the vesicles that are holding the neurotransmitter to bind to the cell membrane, and then they will be able to go through exocytosis and release that neurotransmitter into the synapse. And so those, again, are the pictures of it. The next one is the Golgi. The Golgi apparatus is I have on here, it's kind of like UPS or the Postal Service, but it's really more like Amazon, because it is tweaking or finalizing a lot of proteins or other molecules, and then sorting them out and packaging them up and then mailing them. So it's not just packaging and mailing, it is actually modifying, sorting them into the right package, then putting them in a vesicle, which is their package, and then shipping them to the cell membrane or to another organelle. So it's actually more than UPS or more than the Postal Service. It's doing more of a Amazon type job. It also produces the lysosomes. So lysosomes are really just glorified vesicles that have enzymes in them. It is a flattened stack of membranes. It is also fluid, and what I mean by fluid is not that it's liquid, but fluid in that it's always changing. So it's always got vesicles coming in from other places like the rough ER, and it's always got vesicles going out, pinching off and going out towards the membrane. And so it is always in flux. It is always moving or changing. And so that is how their structures are related to their functions. So the difference between smooth and rough ER and what they do, their location in the cell. If you look here, here's your nucleus, there's your rough ER. It's usually snuggled right up against the nucleus. Your smooth ER, usually the rough and Golgi are near each other because they are communicating, but there's space around the Golgi so that it can pinch off those vesicles as well.